All right. Hi, guys. Good to see everyone. I was thinking are we still waiting for the faculty to join, to other professors to join, or
I think
just,
they'll be joining in as they're getting ready to present. So I think we can go ahead
I
and
see.
get started.
Okay. All right. No worries. Okay. Cool. Okay. So uh, technically, I wasn't really invited for this event, but I hope it's okay. So that I'm here. Basically, I was asked by, and I'm obviously happy to be here, of course. So I was asked by Dr. Westlake to give a presentation on his behalf, as he can't make it. And um, yeah, so I'll do my best because technically I received this literally an hour ago. I know most of the details, like in general, but maybe not everything, but I'll definitely will do my best to answer your questions. Okay, so my apologies if I may miss anything, but it's basically pretty straightforward. Okay, so let me share my screen somehow. Okay, that's this one, right? Okay, so I hope you can see my screen.
Yes, we can.
Awesome. All right, thank you. Nice. So let me put this thing. Okay. Right. So I like to talk about this new opportunity for forensic science lab internships, and in general, as you all hopefully know, internship course, or JS 181, is a required course, one of the key requirements for literally all our programs, both JS and FS. You must complete an internship in order to graduate on time. So you must complete at least 3 units. But as I tell all my students, I highly recommend completing more than 1 session, so more than 3 units, not because of the units, but because of the hands-on experience. So you need as much hands-on experience, as much practical experience as you can get through your studies, because that's literally number one question in job interviews. And the more you get, the more diverse that experience is, the better. Okay, so if you want to secure your dream job in forensic science, that's literally number one requirement. So do your best to complete at least 1 semester worth of internships. And it could be, different place. It doesn't have to be the same lab or same agency, but the more the better. Okay, so now, as you also may know, there's a limited number of forensic science internships that we can offer, although we do offer quite a large variety of options. Those that are offered by crime labs, these options are limited, well, naturally, because of the number of crime labs. And sorry, I don't see the chat, but I see there's something in the chat. But okay, so if you guys have any questions, just to let you know, please feel free to interrupt me. So unmute yourself and ask questions. Okay, so so as I said, there's a limited number of those internships offered by crime labs and biotech industry, naturally, and same goes for internships with the faculty, forensic science faculty, because, well, there are only, well, 3 of us, soon to be 4, but so we can take a limited number of students that we can supervise, and as a result, we were thinking about other options, so you guys can gather this experience, so you can get more hands-on for your career, and of course graduate in time, right? And that's this is the one of those that came up, which we think might be relevant and beneficial for many students. Okay, so what we would like to propose, and this is just, it's not it's still not so we're still not running it officially. we kind of run this unofficially. Myself, I've been doing this for quite a few years, and I have a few students who've been involved in this kind of arrangement, but is an official program we still haven't started, but hopefully we're going to start this this fall. So these are the details, kind of general details so far. Okay, so we talk about an opportunity for students to help with lab-based sections of our forensic science students, to set up the course, to the labs, to clean after the lab, to teach your peers, and as a result get hands-on experience, as I mentioned, in basic lab techniques, in also peer teaching, which are all very essential skills, right? So so you'll essentially will be doing your internship as a TA, as a teaching assistant, helping with various lab courses based on availability and or essentially program needs and your capabilities. Okay, so specifically how that's supposed to look like. Well, if you're interested in helping with the lab as a TA and getting credit for that experience through your internship, so JS 180, 181, you would have to meet with your lab instructor, with the course instructor, to make sure that you are aware of that course needs, what needs to be done, what kind of consumables need to be prepared, what this lab is about, and all these kind of things. Usually that would be pretty straightforward, because most instructors would require you to first complete that course, so you would already know what this course is about and what this lab's about. Okay, and then you'll be required to help with the lab prep, right? As you all did, different lab courses, 162, 167, 57, and others. There's a quite substantial lab prep for those courses. Okay, on top of that, you would have to be there during the lab, most of those labs, to help during the lab, to provide support for instructors and help students in their lab activities. And however, you as undergrads, you won't be doing any grading, because that would be considered an ethical issue, right? So that's unfair, right? So we want to avoid all these kind of things. Okay, so it's mainly this technical support. However, you would need to hold some office hours. I'm not sure how long that would be, probably an hour or two during the week, but we're still working on that, so that's to be detected. And then we'll during these hours you'll be helping other students with their questions regarding the course. Okay, so in terms of application requirements, how we're going to work do these. So as I said, we still haven't finalized this, but hopefully soon. So by, I guess, this summer, we'll have specific procedure, and we'll obviously advertise this. We'll share this with all these students. The application will need to be submitted online. Specifically for the dates, we're looking for around June for the fall semester applications, and if you want to be considered for the spring, you need to submit your application around October. Okay, so now very importantly, students would have to demonstrate that they successfully completed that course that they are applying to be a lab assistant for, right? So for example, if you completed whatever, I don't know, FS 162 with A or A plus, then you have a good chance to get the spot and be considered to be TA for this course. And there might be some additional requirements that we are still discussing, and of course everything will be advertised by this time. Okay, now obviously the number of those internships is limited based on the program requirements based on instructor and course needs. So if you apply, the spot is not guaranteed, unfortunately, but that's pretty much the same as with any other internship. But we'll do our best to allow as many spots as possible. Okay. Oops, that's already about my thing. So before I go there, that's really briefly about this whole arrangement. So if you guys have any questions, please feel free to unmute yourself or type in the chat, whatever you prefer. And then I can talk more about specifically about internships that I offer in my lab. That's on top of TA or this lab assistant, this arrangement, and then my colleagues will also talk about opportunities in their labs, in their groups.
I had a question.
Sure.
How recent did you have to pass that class? Can it be like a year ago or how?
Well, it would depend on the particular instructor. I can talk about myself. So, for example, when I consider TAs for my courses, and usually that's FS 57, I would need you to complete that course, last semester or even 2 semesters ago. So from my perspective, the more knowledge you have, the more experience you have, the better, right? So it should be minimum like last semester. So you complete that course, but if you complete like last year, that's fine with me. I don't have any problem with that. Yeah. So also, like, some instructors may want to to be familiar with that specific student who applies. So like, that you were a student in their section, and some may not have that preference. So if you completed that course in someone else's section, that's also fine. It really depends on the instructor, I guess. So I hope that answers your questions or question.
Thank you.
All right. Any other questions? Again, I mean, this whole thing hasn't been finalized, but that's like basically that's how we see that. And as I said, I've been doing this for quite a few years, and I had quite a few students who were helping with with lab setup, and it's been a great experience, hopefully for both of us, at least for me, that's for sure. And then in most cases, I also tend to involve those students in ongoing research in my lab. Okay, so if they have time for it, and if they want to be involved, it really depends on their availability. Okay, if they want to be only a TA, that's fine with me. Or if they want to be involved in research, that's also fine. Okay. But again, as I said, it really depends on that particular instructor. If they have availability in their lab, if they have time, of course, then I guess that could work this way as well. Any questions? Okay. If you'll have any questions later, that's fine. I'll be here for the next probably half an hour or so, but you can later just email me or other instructors, and we'll be more than happy to answer your questions. Okay. So I guess for the sake of time, let me just briefly go through research internships in my labs, which I mentioned. That's not a possibility. That's more like a traditional possibility, if you want, option for students. Just very briefly, for those of you who are interested in research internship. So here are the options in my lab, and basically these are the these are the my research interests currently. So I'm running several research projects in parallel. Some of those are focusing on what is called DNA transfer. So as I hope you know, my area of expertise is forensic DNA analysis, but also interested in some other areas in forensic science and beyond that. And essentially, I'm looking into these aspects of secondary and tertiary DNA transfer and its implication into whole criminal justice, if you want, and then looking into forensic DNA analysis as a tool for forensic intelligence, something which you may use for what's called cold cases or missing person cases to help with providing this forensic intelligence, for example, about persons' physical or visual appearance, their facial appearance, their pigmentation, eye, skin, hair color, and other traits, to help with identifying those missing persons or providing some intelligence in those cold cases. And unfortunately, there are over 250,000 such cold cases in the United States at the moment. So that's a huge number, and many of those do have some biological evidence that could be could provide a lot of hints and clues to help with solving those cases. Okay, and finally, I'm also working on other research topics around forensic genetics and forensic DNA markers. I'm also using machine learning and AI to help with generating a pipeline of predicting visual appearance from DNA sequence with my colleagues. And as a result, I do take students which are mostly forensic biology majors or sometimes forensic chemistry, sometimes digital evidence, and also computer science students from College of Science who are more focused on the machine learning part of this project. And our forensic science students are mostly working in the lab, doing wet lab work in my lab. Okay, so in terms of experience or this whole overall kind of arrangement, if you want to apply to join my my research groups, I would need you to essentially commit for 2 semesters or ideally a whole year, and that's because it just takes a lot of time to learn the basics, and until you learn that, the semester is over. So yeah, so most of my students are there for a year or at least 2 semesters, and then you would need to complete at least by 30 and ideally FS 57 as well before you can apply. I also have, as all of us, a limited number of spots, unfortunately, so I can supervise only a few students. It takes a lot of time, and I can't take too many because that wouldn't be just unreasonable and also unfair for the students, because I won't be able to provide enough time and for this shit. So yeah, so that's, I think, briefly about my internships. If you're interested to join my group, please send me an email, or even better, just drop in during my office hours. That's Monday, Tuesday, 4 to 5 p.m. on Zoom. You can find the Zoom link on the web, or I'll post it later. And yeah, I think that's all. Wait, do you guys have any questions?
So in order to apply for that lab research opportunity that you were talking about, you just want us to like drop into your office hours, or is there an actual link that we can like apply to?
Well, so currently we don't have a dedicated website where you can apply. We may have, maybe my colleague Dr. Bolton will talk about this, so we may have something like that, but I'm not sure if it's already in a working state. But specifically for my research group, you would just send an email if you're interested, ideally with your unofficial transcript, and then we can discuss this more further during my office hours.
Okay, and just the prerequisites that you said Bio 30 and FS 57.
Yeah.
If we've completed other biology classes and chemistry classes, but are going to take FS 57 next semester, would that kind of weed us out?
Not necessarily. It really depends. So if you are taking FS 57 in the same semester, that might be okay. Again, keep in mind that I have, as all of us, have very limited number of spots that we can offer. So like, for example, currently I have literally no spots, but I may have some this upcoming fall.
Okay.
Because quite a few students are graduating and all that, but it really depends on its availability. So don't be shy. Just send me an email and we can discuss this further.
Okay, and what's your email?
Oh, I'll post it in chat.
Okay,
Yes.
thank you so much.
No worries. Any other questions before I pass the mic? That was supposed to stop my intimidating presentation. Okay, so hold on. Okay. I'm not sure why it's still going. All right. Yeah. So do you guys have any? I didn't see the chat. Okay, there's nothing in chat, but any questions? Yes, exactly. So please post in the chat, and I'll just reply in the chat. All right. Okay. So for the sake of time, I'll just pass the mic to my colleagues. I'm not sure who's next. Is it Dr. Warren or Dr. Bolton?
Dr. Bolton will be presenting next on her lab research.
Awesome. Okay. All right. Well, thank you so much, guys.
Thank you, Dr. Barash.
All right. Well, good afternoon, everyone. Hope all is well on your end. I'm Dr. Shawna Bolton. So I'm going to talk a little bit about the research project that I've been doing, how to go about applying for it. I did not get a chance to create a PowerPoint for a second internship that I've got going on in the background, but I'll just do a brief little chat about that as well. Do I have the ability to share my screen with you guys?
Should be available.
Okay, awesome. So I'm going to go ahead and share screen. Okay, hopefully you guys are able to see that. So now I'm going to go ahead and bring up this PowerPoint presentation. Okay. See? All right. Okay. So back in 2017, when I actually started here at San Jose State University, I was also doing a bit of advising, and internships have, for I think several years now, been a part of our program experience. I think they're incredibly wonderful. Simply because when I was doing an undergrad at the University of Toronto, we did not have that kind of experience or hands-on experience to go out in professional field and see how it all connects with the way that we're learning about the theories and the practices in forensic science as you guys are doing today. So the research that I've been doing pretty much looks at cartilage, at least postmortem cartilage, and what's happening to it when it breaks down from a biomolecular standpoint. So really, and truly why I'm doing this is I'm trying to figure out, can we use cartilage as a forensic tool to help us with investigations? So I'm going to just briefly talk a little bit about what exactly is forensic taphonomy, because so Dr. Warren and I have created a forensic taphonomy class. So if you're not aware of it, it's there, it's available, and you're more than welcome to take it if you've got space to do so. Then I'm going to talk a little bit briefly about death investigations, how the research that I'm doing pretty much ties into that, and the research methodologies for this particular research project that I'm doing. And then last but not least, I'll tell you guys how you can go ahead applying and when to do so. All right. So forensic taphonomy. How many of you guys have actually heard of that terminology before? And I'll just get you guys to give me a thumbs up. Of course, Dr. Warren. All right, so we got a few thumbs going up. That's pretty good. I'm glad to see that. Typically, when I talk about forensic taphonomy, people are like, what the hell? Like, I have no idea what you're talking about, Dr. B. So I'm pretty surprised to see that. So that being said, whenever we talk about I want to go first, take a step back. I'm a forensic anthropologist. And so we tend to use our knowledge of the modern skeletal system, and we're going to try to extract as much information as possible from human remains. And that's simply because we're trying to pretty much help identify a victim. And we want to try to reconstruct the events that surround their death. And so we work to create a biological profile that consists of information about an individual's sex, their age at death. We're also looking to possibly tell what their ancestry was. Um, we're looking at stature as well, how tall they might have been at the time they died. And we're also looking at things such as individualizing traits, simply because we were trying to narrow down the possibilities of who our victim or suspect might be. And so the study that I conducted at present pretty much involves taphonomic research. So whenever we talk about forensic taphonomy, this is pretty much the study of postmortem remains and their fate. So what happens to the body when it's deposited in its, I want to say in situation, right? So wherever it's deposited, whether it's above ground, below ground, or in submerged situations. But we also want to know, how does the body impact the environment that it's deposited in? Right. So at the end of the day, when we think of forensic taphonomy, it's pretty much from the word taphonomy, which is the law of burials. Right. And so, for that being said, it involves investigating soft tissue changes, the rates of decomposition and the patterns in which decomposition happens. We're also looking at the dispersion of body parts over time. And we're also looking at the modifications of soft tissues and bones. And so, whenever we talk about forensic taphonomy, there's actually two branches of forensic taphonomy. These are actually known as geotaphonomy and biotaphonomy, and I'll talk about that on the next slide. All right, so when we talk about geotaphonomy, geotaphonomy is pretty much going to be the study of how the immediate environment impacts human decomposition, but it's also how decomposing bodies affect changes within its surroundings. So, for example, some of the stuff we would study are soil disturbances. We would look for footprints that are found at the bottom of a grave, if in case we suspect that there was some sort of illicit activity that happened. We're also like looking at the changes in the soil's pH, right? So typically this is going to be below bodies that are decomposing. So on the other hand, we also have what's known as biotaphonomy, and this pretty much examines the remains themselves, and it pretty much asks how decomposition and destruction of the hard and soft tissue came about. So this is the branch that my research pretty much falls under. And so biotaphonomic processes, they're pretty much going to be influenced by environmental factors. So for example, things like climate and animal activity. But they're also impacted by things like individual factors. And these are going to include things such as a person's body size and their age at death. But there's also this other aspect known as the cultural practices, right? So for instance, whether or not a body was embalmed, and so what are some of the autopsy procedures that have taken place that might impact soft tissue decomposition or just decomposition in general. So most of you guys are probably well aware with forensic science that there are various methods that we use for estimating postmortem interval. So before I go into all of that, I just have to briefly explain what exactly is postmortem interval. So postmortem interval is pretty much the amount of time that has elapsed since a person who's been deceased is discovered or essentially about to undergo examination in the medical examiner's office or coroner's office. Right. And so that being said, we typically rely on things such as skin pallor, right? So these things are pretty much going to be different physiological changes that happen soon after a body starts to essentially undo itself. So this is a process where we're talking about decomposition. The thing that I want to talk about briefly when we're talking about postmortem interval is that you want to bear in mind that this is a very, I want to say it's an estimate, right? That's the key word. We are not necessarily going to have spot on responses to let you know that, hey, well, this person is exactly how many X hour or our hours dead, right? So at the end of the day, we're essentially just trying to give you a time frame. But a lot of the time frames that we're using are pretty much limited. So once these physiological changes are over, they're useless. All right. And so that being said, the best we can do with pretty good accuracy using soft tissues is up to 100 hours after death. So that being said, these hours are typically considered the first of the early phase of postmortem indicators of time since death. And first and foremost is we have skin pallor. And so skin pallor is something that typically is going to occur within 15 to 30 minutes after a person dies. And it's going to be marked by a lightening of the victim's skin tone. And this is on account of the fact that blood is no longer circulating through this person's body. And so this method is typically only going to be useful within 30 minutes of having died. So the other method that we have is livor mortis. And so livor mortis is pretty much when the lower portions of the body turn a purplish color on account of the blood settling in the direction of gravity. And so once the blood stops flowing through your body, it's pretty much going to settle. So lividity starts virtually immediately, right? So although this isn't always noticeable right away. So on light-skinned people, we tend to see what is going to look like a pinkish color, and it can typically be seen within an hour, but darker skin tones can hide lividity, right? So we're going to have to sometimes depend on other methods to assist with that. So within 8 to about 12 hours, this is when lividity is typically going to become fixed. And we see a bright red lividity called a cherry lividity on a body that's found typically either indoors. And it can be a warning sign for the fact that a crime scene investigator that's there, they might actually stop and have to say, is this carbon monoxide poisoning, right? And so that's something that if that ever should happen, you should be evacuating. So another method that we tend to use to try to estimate time since death is rigor mortis. And so rigor mortis is where like muscle proteins, such as actin and myosin, they're pretty much locked together and they're going to create a stiffening of the muscles shortly after death. And so this stiffness becomes apparent typically within 1 hour in the smaller joints, such as things as your fingers and your wrists. And then pretty much rigor is going to reach its maximum at about 12 hours. And so the body is going to remain stiff after the 24-hour mark. So all rigor pretty much disappears after about 36 hours when the body begins to decompose further and proteins that make up our body, they pretty much are going to begin breaking apart. So rigor mortis is greatly affected by temperature. And so higher temperatures can actually accelerate the onset and passing of rigor mortis, and colder temperatures can actually retard rigor mortis. And so that being said, another method that we use is algor mortis. So algor mortis typically is going to refer to internal temperatures of the body after death. And so the temperature of the body typically cools down at approximately 1.5 to about 2 degrees Celsius per hour for the first 12 hours. And so after this point in time, it's usually going to decrease to about 1 degree per hour after that. And so this method pretty much assumes the environmental temperature is lower than the body temperature. And so the internal temperature of the body is typically measured from the rectum or inside the brain. So essentially shoving something pretty far deep in the nasal cavity. And then the single most accurate method that we do have today using soft tissues is essentially the potassium content of the eye's vitreous humor, which shows typically a linear rise in potassium ion concentration. And this happens within a time frame of 12 hours to 100 hours after death. And so that being said, after death, blood cells in the body, they're typically going to break down and they're going to release potassium. And so in the eye, this process happens a lot more slowly and more predictably than it does for blood. And then when we have no more soft tissue, I'd say soft tissue physiological changes to look at, forensic entomologists are the ones that we pretty much depend on. And so that being said, we're looking for insect activity to determine the approximate time of death. And so we know a thing or two about this. We've heard, hopefully, in our forensic classes about flies and their life cycles. So we kind of use that information to help us assist with time since death. And so we go from that and using insect succession to hopefully estimate time since death anywhere from 3 to 13 days after death if we're dealing simply with flies, but up to 35 days if we also have beetles that are present. So that's what we typically use as forensic, I want to say, forensic investigators. However, although a lot of these methods that I've presented are considered some of the most valuable methods for estimating postmortem interval, especially with increasing time, the accuracy of these methods pretty much dissipates and becomes less reliable, and it makes it difficult to estimate postmortem intervals for cadavers more than a few days old or bodies that are not necessarily accessible to insects. So these would be situations where bodies are typically buried below ground if they're submerged, if they've been wrapped tightly in clothing or some sort of cloth material. And so we really can't rely on these methods. And so the research that I've been conducting actually explores the biochemistry of postmortem cartilage in order to understand the mechanisms that are involved in its destruction, but also to determine whether or not this tissue can actually be used to estimate a postmortem interval. And so this, again, is just the period between physiological death and medical legal examinations of a body. So there's not been a great deal of research since I started this project back in 2010. There's very little that explores cartilage as a forensic tool. So back in 2002, it was actually individuals that were conducting research, and the individual is named Leskowski. And so they actually noted that there was a correlation between chondrocyte loss and postmortem interval. And what I mean by chondrocytes are pretty much just the cells in cartilage. That's what we call them. And so they were noticing that as postmortem interval increased, the percentage of viable cells decreased with increasing postmortem interval. My predecessor to looking at cartilage was another PhD student at the university that I attended in the UK, and his name was Rogers. He actually noticed and characterized the systematic changes that cartilage underwent at the macroscopic level. And so what he did is that he actually highlighted changes in color and diminishing robustness of the tissue, but he also noticed microscopic changes, things like loss of nucleic material over time. And so that being said, a lot of the times my research is pretty much one of the first to look at the biomolecular breakdown of cartilage from a postmortem perspective. So why did I decide to look at cartilage? Well, for one, it's pretty much a suitable candidate for postmortem research, and that's because of its physical properties. It's pretty much situated on the articular surfaces of bone, and it's actually protected by a membrane containing synovial fluid that is inside of a joint capsule. And around that joint capsule are other surrounding soft tissues that offer it protection from the immediate environment. And so whenever we talk about cartilage, pretty much it's composed of what is called a cellular matrix. And so that is what I just made mention. We're looking at chondrocytes. And so that's these little cell materials in this region, just to give you a demonstrative diagram. And so chondrocytes are what are pretty much responsible for maintaining the tissue, but also destroying the tissue. But then we have all the stuff that are around the actual cells, which is called your extracellular matrix. And that is pretty much formed by a network of things like collagens and proteoglycans and, for instance, non-collagen proteins. So that being said, Aggrecan is usually considered the major structural proteoglycan that's embedded within the extracellular matrix of cartilage. And so they're like beams in a wall that are going to give that cartilage structural support. The thing that's pretty awesome about cartilage as well, why I looked at it, is that it has a low cellular density, which means that it grants cartilage a prolonged resistance to a lot of the degradative fluids that are generated during the processes of autolysis and putrefaction. So at the end of the day, I'm kind of baffled that no one ever thought to look at it. So yeah, these are just pictures of the major structural components of cartilage and so forth. And that being said, just to give you guys a little gist of what we do. So we do have a body farm, I like to loosely call it, because it's a really tiny, tiny body farm that's located behind the music building. So that's just in front of the student union. So there, if you're ever walking behind the music building, you will see a tall black fence. That is us. And that's where we essentially conduct our postmortem taphonomic research studies. So for my particular research, what we do is that we have two different types of projects going on, or experiments, where we're looking at what's happening to cartilage in a buried environment versus cartilage in a submerged environment. And so typically that's going to involve us gathering quite a lot of trotters. And so that means we typically have about 6 control samples, because that's what you would use to essentially work on and practice your techniques and your skills. But also because we want to use our control samples for comparative stuff. And so that being said, we typically take 18 trotters and we would bury or essentially create 6 graves, and you would put 3 trotters per grave. And then what essentially happens is that every week we go and we unearth a single grave. And then we dissect our trotters for cartilage samples at specific joints. So that being said, we also take an additional 18 trotters and we divide them up amongst 2 bins with water. So we're pretty much dealing with stagnant tap water, just seeing what happens to cartilage in that type of environment. So some of the things that you guys will learn with me, if you decide that, you know, you're interested in this particular research project, is that you will have to learn how to extract protein. And we sometimes will have to end up using protease inhibitors, and these are things just to prevent enzymes that are naturally occurring in our bodies from further breaking things down. Sometimes we will do a digestion, but that really just depends on whether or not those of you who decide if you want to take on this research for a second semester, whether or not we'll be doing immunohistochemical stuff. And then we pretty much do SDS page, which is sodium dodecyl sulfate polyacrylamide gel electrophoresis. And that is an incredible mouthful to say. But the whole premise is very much the same for those of you who loved DNA analysis, where we're pretty much taking our protein samples, loading them in wells, and we're using a powered system that pretty much has an electric negative or electronegative node at the top and a positive node at the bottom. And we're running an electric current through our samples. And what we're hoping to see is that we get a separation of protein fragments. So that essentially with the electric current is what's essentially separating things based on having a high molecular weight. Those things will stay closer to the top. And anything with a really low molecular weight will be able to be pulled further along in the gel. And so typically this is what we end up seeing is some sort of banding where we have our separation of proteins. And then we essentially do Western blotting for those, again, who are interested in coming back and learning a new technique, is where we pretty much focus on specific target protein that we're interested in looking at. And then we add antibodies and pretty much try to get a visualization of what's going on over the 6 weeks that our samples have been buried or submerged. All right. And so that's pretty much what we're talking about when we're talking about Western blotting. So first we do a vertical separation of our protein fragments, and then we're going to transfer these proteins horizontally. So that means from here to here um, with the electric current. And then once we place those proteins on a membrane, then we are essentially attaching protein antibodies and highlighting them and trying to get a visual on X-ray film and so forth. So the other aspect of this project is that we are also doing live dead cell assays. So that means some of the stuff that I did previously is look to see where along the cartilage's surface is going to be considered the best representation of having the most live cells um, shortly after death. And so the area that I found was actually this inferior groove in the knuckle or the metatarsal joint. So that's basically this region here on our pig trotters. So you guys would be essentially assisting with this, where we would stain thin sliced sieve slivers with so calcium AM. So that is a probe, a fluoroprobe that highlights all the cells that are alive. So if your cell membranes are intact, basically you're going to stain the cell membrane green. Anything where the cell membrane has essentially been destroyed and the nucleus is exposed, propydium iodide is going to pretty much highlight those dead cells, which is essentially highlighting the nuclei. So then we pretty much would look to see and essentially be counting the total number of chondrocytes or cartilage cells that we're able to see. And then from there, we would count what's alive and then try to generate some sort of graph looking to see what happens over time with our live cells, bearing in mind that we have to take the ambient temperature virtually every day to make those comparisons and see what's going on. So typically, if you're interested, I usually take applications about a week prior to registration. And so you can pretty much go to the Justice Studies webpage. You can click on current students, and I'll probably just quickly show you guys. Dr. Barash, did you happen to do that already?
No, so feel
Okay.
please, yeah.
All right. So let me see if I can just get rid of this, and I'll show you guys where to go to apply for all of our internships as with faculty. There's a pointer. Okay, let's get rid of that. All right. So this is where you could pretty much go. I'm going to take you guys to the Department of Justice Studies. All right. This is where you'll click or hover over current students. Click on undergraduate, and you're going to scroll down to forms. Click on the download forms link. You can click on JS 181 internship, and it'll take you right here. The form that you are most interested in filling out is going to be the one that says alternative internship application. So if you click on that, you'll see that there's a JS 180, 184 ad code application. You want to fill out your name and details. And so for my particular internship, I always say to you guys, just tick off the first box that you're applying. Technically, I don't know why they're calling it JS 180, but it should be JS 184 because it is a directed study with us. And you're doing it to satisfy your JS 181 internship. And you're going to take all the reasons that you're pretty much doing this alternative. Typically, I'll just tell students, highlight the one that says equivalent experience working with faculty. Now, you also want to make certain that you read this little area because sometimes students forget that. You do need to attach a separate letter just outlining or explaining why you're interested in essentially doing this alternative internship rather than the traditional JS 181. All right. So it could be that you're interested in our research. It could be that this is a more convenient way for you to get your internship experience because traveling to and from, say, another internship experience elsewhere is incredibly difficult for you. All right. So whatever the reason is, just make sure you include that. You will essentially submit that to me. For me, per se, let's see. There we go. I also include a Google form that once you've initiated the process of applying, I will submit that to you guys. And then I have you guys pretty much just go ahead and um, fill that out. You would include your resume and so forth and your transcript. And then, yeah, just, how you heard about this internship, your dates and times for availability. So for me, I'm typically flexible with regards to hours. I usually have a specific couple hours that are dedicated to me giving instructions, as well as training. And so after that, the expectation is that you come in at least a minimum of 8 hours per week to at least get through the 120 hours. Although, I always say that's the minimum for the research that you guys do with me, because chances are you're going to end up going over that time period trying to get results. So that's virtually all that I have. And I guess the other aspects, I'll just quickly go through here. So the other research experience opportunity that I have is with Alliance for Hope International, which is our collaborators. They pretty much have joined forces with us here at San Jose State University to enlist our help with reconstructing crime scenes. So that's using 3D um, software. And that's just in hopes of revisiting cases that they're actively investigating where they believe the victims are not necessarily, their death is not necessarily the cause of a suicide, but has been masked as that. And so we actually just try to go about helping them answer some questions about their deaths or just creating 3D renditions so they can give people, their juries, a view of what the crime scene looked like without it being too gory. And just to give you guys a quick view of what that looks like, let's see if I've got that here quickly, and then I'll take questions. All right. So here, and I'll put that on the next slide. Okay, let's just quickly pull this over here. Is it going? There we go. So this is an actual case that like a previous student worked on for an individual by the name of Libby Caswell, who was suspected of essentially being strangled to death, but whose death was made to look like a suicide. So we kind of do that kind of stuff. So if you guys are interested, feel free to reach out to me. Do you guys have any questions? And I see that there might have been stuff in the chat. Okay. All right. Well, if you guys don't have any questions, you can find me on the Justice.
Sorry, Dr. Bolton, we did get a question about if there are any prerequisites for your lab or internship.
So typically for any internship, you also need to have completed your JS 100W or your FS 100W. So for me, that's really all that I require. For me, this is meant to be more of an experience where you get to do a little hands-on in the field, but also in a lab setting. So hopefully like the stuff that I do ties in with what you're learning in your forensic bio or chemistry classes. Um, but also for me, it's more just to give you guys the opportunity to realize this is something you never thought of doing, or taking, things further with your education, just to give you guys the confidence. So a lot of the things that I do actually utilizes the UK way of learning, where it's here's your instructions, and then it's very independent. Go figure out what's going on, and when you're not able to find answers, come and see me. Right. And then we'll kind of try to talk things through. So it's more giving you guys the confidence to troubleshoot, right? And so it's also being confident working in a lab and being confident that you can take on a graduate degree. So I know I didn't really tap into, but I've had quite a lot of students that have essentially gone on to work with crime labs. They've worked for the Department of Justice, the FBI, and biotech companies. Right. So they've always told me great things. And I've also had students go off to do their masters and some directly to doing their PhDs that essentially wrote me a message and said, "Hey, Dr. B, thank you for this experience. I'm ahead of the game where a lot of my peers don't know what they're doing in a lab." Right. And so that's what the hope is that you guys recognize you can go out there and be these professionals as well. All right. Were there any other questions? No. Okay. Well, if not, I am done blabbering. I'm going to pass the mic on. I believe it is Dr. Warren. I think you might be next.
Thank you, Dr. Bolton.
You are welcome.
Hi, everybody. I'm going to share my screen then. Okay, I had to, put some ugly bugs on there. Okay, so for my internship, basically, what I want you guys to do is just to send me an email if you're interested. So I'll start with that. That's the absolute best time to contact me is just send me a quick email because that's how I get everybody just who says I'm interested. I'm like, send me a quick email and that will remind me to send you paperwork your way, and I'll say, okay, let's fill it out and get it back to me and so forth. So that's the absolute best way to get started. But let me just tell you guys first a little bit about my lab. Okay, so forensic entomology, this is what I'm gonna just have outlined this here. What is it and why use it? Then I'm gonna go into a little bit of cases. I had to delete some of my cases because this is being recorded. And just because I can't show you, can't have recorded crime scene photos. So for casework, we go into homicides. We do neglect and abuse cases. So that could be animal, it could be elder abuse cases, like in like an elderly home. And it can be child abuse cases. So think of it that way. So it's not always someone is deceased. And then I'm going to go into the internship and a little bit of current research. Okay, so just close this off here a bit. And I'm not looking at the chat, so if you guys want to even just interrupt me and say, Jodie, blah blah blah, this is my question, go for it. So historically, forensic entomology, people think it's just a brand new science. It really isn't. The first case that actually was done was back in China in 13th century, so it goes that far back. Then we see it kind of pop up again in the 1850s in Europe. Then again, Britain in the 1930s. In North America, we actually started using it really here in the 1970s. The American Board of Forensic Entomology, which is the one that now all the forensic entomologists, when they get board certified, they join. And that started in 1996. I'm not going to go into detail about those because we don't really have the time for that. But just so you're aware, it's not a new science. It is actually quite old and has been used for quite some time. So we do cases here in the Bay Area. Dr. Honda, Dr. Jeff Honda was doing them cases a long time ago before I even moved here. The last cases he worked on actually was 15 years ago here in the Bay Area, and he worked with Santa Clara County Sheriff's Office. He worked with the San Jose Police Department, with the DA's office, and Public Defender's Office. So really, I need to get the next steps I really need to do. I've been trying to collect more data just to really apply my own data to the area. But the next steps really are to train current officers and death investigators. So by having a forensic entomology class and a taphonomy class, I've been trying to I basically have been training more crime scene investigators, which is you guys, so that when you go to a scene what to do, what to collect, who to contact if you need to contact me and say, Jodie, get your butt out here. We need to look at a case, or hey, I've collected these insects. What I'm going to send them to you, that kind of thing. Now actually one of the biggest things we do is we look at time since death, which is basically the postmortem interval. And we always think of it in terms of, homicides. Well, it's not always about homicides. When we think of time of death, we think, first of all, probably everybody here has lost someone or a pet or something. You probably grieve in terms of time. Oh my gosh, it's been, I can't believe it's already been a month since, Muffy died, or, whatever it might be, or since, grandma passed away. So we grieve in terms of time. So to be able to like heal and to be able to get past, to get beyond that grief, you never really, get over it, but you like to be able to, go on, you need time. And so we grieve in terms of time. So we need to know details about a case. We need like, if you've ever lost someone, you didn't know the details about what happened to them, it's the most devastating thing ever. So to know like all details about what happened to them is very helpful. Even if they're terrible, it helps. So to be able to say, okay, it's been such and such amount of time, so that this is very helpful. And they died at such and such a time. And to be able to say, okay, yeah, I can't believe it's already been, 5 years, because it does go by pretty quickly. Then you're grieving, and it's the time just helps. So I just wanted to make sure that you guys are aware it's not always in terms of like a death investigation. I mean, it can be a death investigation, but it's not always in terms of a homicide. But in cases of homicide, we can use that information to confirm or deny a person's alibi. We can use it to exonerate the innocent. It can help us to identify a body. We need to have an idea of who a person is before we can, start doing DNA or fingerprints and that sort of thing. So, like, say we have, two little girls have gone missing, and we come across human remains. It looks like a little girl. Remember, as time goes on, decomposition, it's hard to tell. We can say, okay, well, Jessica went missing 5 weeks ago, but Jennifer went missing only 2 weeks ago. These remains have been deceased for 4 weeks at least. Then we can say, oh, well, it's definitely not Jennifer, but it could have been Jessica. That could be Jessica. So we can now focus on getting all of her dental records, her fingerprints, that sort of thing, and start to actually examine and see if that's her. And so in the same way, we can point an investigation into the correct time frame. So we can put all of our resources into that time frame of when we want, like, last time the person we saw so someone might be like a bit of a oh my gosh, I'm not sure what the word I'm looking for is, but maybe they're nobody's really seen them around lately, but the last time anyone's really seen them was, say, 5 weeks ago, but their death may have been 3 weeks ago. We want to make sure we point all of our investigative tools towards 3 weeks ago, not 5 weeks ago, because otherwise we're wasting resources. So in that sense, that's what we want to do with knowing time of death. So my lab really focuses on time since death. We do more than that, but time since death is huge, and the insects in the lab basically is what helps us do that. So we use two main methods in forensic entomology. The first of these is looking at the immature blowfly development, and this is one that I focus on in my lab. And I'm going to talk about that in a second. The second method is successional ecology. So the first one is looking at insect development. So what stage has the insect reached in development? Because the insects are developing on the dead body. So if they've reached a certain stage on the dead body, then we know they've been there for that length of time. For the second methods, we know that certain insects come in a certain sequence. So then we can say, okay, well, insect A has been there, insect B is here right now, and we know insect C is about to come. So we know then how long it takes for that's predictable sequence to occur. So we can provide a conservative postmortem interval estimate, basically, is what's happening there. So for that first method, we're looking at time since death, and we're looking at the immature development. So this is your basic figure of what happens in immature development. Dr. Bolton showed you basically the same thing. The adult fly is going to come lay eggs. Those eggs are going to hatch in a first instar, and those are going to molt in the second instar. They molt again in the third instar. At a certain point, third instar is the voracious feeders. They're the ones that really break down a body, and they're going to just keep feeding and feeding and feeding, and they really clean off the body really well. At a certain point, those insects stop feeding and they wander away from the body. And that, when they do that, that's the post-feeding stage. They look identical to these guys here. So they pretty much are the same. But they just, all of a sudden, they look the same, but they just now have just done this. It's more of a behavioral change. That's all it is, a behavioral change. And then they will shrink on either end and kind of shrink into like a football shape. And inside, everything then turns into a white goo. Everything just dissolves. And within that cocoon, basically, it's just like a caterpillar and a butterfly. Everything reforms inside of that. It's a puparia. Everything inside of that is going to reform and form an adult blowfly. And that is then going to hatch and break open the one end and emerge as an adult. So that's your life cycle. So that's what we do in the lab all the time. So if we come across now human remains, we want to get the oldest stage on the body. So it could be any one of these here that we'll see associated with the body. So say the oldest stage is third instar, this one right here. We get the temperature records for whatever temperature the body's been sitting at. So whatever those insects have been developing at. Now, temperature, of course, is not nice. It doesn't just stay at a nice constant in temperature. It's fluctuating. So we'll get that information. We want to know what species that oldest insect is. And so we figure that out. Then we consult our developmental data that we've collected. And we do all this collecting, and we do this all in the lab. And this is what you guys do as interns. So we collect these data and we publish that kind of stuff. And so we consult it and we'll say, okay, to get to this stage on this body, at this temperature, it takes this species this amount of time. So that insect has been there for at minimum that amount of time. So that person's been dead for at least that amount of time. They could have been dead longer, but they couldn't have been dead less, because that would mean the insects were there before they died.
How many species do you work with in your lab?
In my lab, well, I usually try to keep it so that I only have a maximum of 2 species. So usually I send out students and we'll go collecting, and depends on what time of year also I'm collecting. Usually what happens is I collect and I get a wild cage. So right now I've got a wild cage, and my students are going through and IDing flies, and they are so it's actually quite a process to ID the flies. And we're separating out Lucilia sericata right now, and that's one main colony I have right now. And we are looking at those ones, but any other species, then they're getting separated into another colony. And usually what happens is, if we keep the colony long enough, they kind of start to break down, so that one colony kind of takes over. So currently I have one colony right now, because my other colony died off. Just after a certain amount of time, they just kind of die off, and it's very frustrating. But I have to always refresh colonies all the time. So I want to go out and trap more and bring the same species in. And if I'm lucky, I'll get the same species, but it depends on what I can catch. So the most I've had, like in labs and stuff, probably has been like 3 different species. I try not to do more than that, because there's a good chance you could cross-contaminate the species, and I don't want to do that. I want to keep them separate. But right now, I currently only have 1 species.
So you only work with blowflies or any other insects?
Only blowflies. in the lab. When I'm doing outdoor research, I will work with whatever is collected. Like I have like preserved specimens of everything. But in the lab specifically, I only want blowflies, mostly because like I don't want to bring in beetles or anything like that, because if they get loose, they could kill my colonies and things like that. So I don't like to bring in anything else.
What does outdoor research entail?
Usually it could be pigs, it could be for the most part, it's putting out pigs. Like I've done that with grad students. I'm trying to set up rats. That's not going to happen now, probably until the fall. But it's just a nightmare to try to be able to do that. That's in forensic taphonomy. So if you want to take forensic taphonomy, I highly recommend taking it in the fall. It's basically so anytime I can, I would like to lay out like whole carcasses and to be able to do that kind of research, but that I basically need a grad student to do that kind of research. It's just too much to do. So that is more graduate work, because it's just too much collecting and so forth with that kind of work. So I've had a grad student do that kind of work before. I would love to set it up for one of you guys to do that graduate work afterwards. So if anybody's interested, contact me. We can do a interdisciplinary graduate work. Okay, so any other questions?
So when are you going to expand to perhaps 2 species or another colony again? Or how long does it take to grow or keep stable that colony? Like how you said they die off at a certain point.
Right. Yes, absolutely. I'm probably going to do it again, maybe towards the end of this semester, maybe start doing that. But right now, the research I'm doing in the lab, I only want one colony because we're focusing, like the internship stuff that we're doing right now, really, it only benefits me to have one colony right now. Just because, and it'll become clearer for what I'm doing, because I'm kind of testing one species at a time to get data for that one. Usually when you publish on one species at a time, or you'll do a little bit of this species, a little bit of that. So like I've already collected some data on Califera, oh my gosh, Latifrans. I've collected some information on that species. Now I want some information on Lucilla serracata, and so it's okay. Like I don't really want the other species, so I want this species now. So it's kind of helpful that that I only have one species going at a time. Multiple colonies of the same species is very helpful. So the interesting thing too is that one of the, or what I should say is one of the biggest factors that affects this kind of research is that we cannot, like like for example, I mentioned Lucilla serracata. It's found everywhere in North America, and in fact, almost like on every continent, basically. We cannot apply data from somewhere else to here in San Jose. So I have to collect data for that species here in San Jose. And the reason is that any kind of Lucilla serracata, let's say up here in the north of California, develops at a different rate than it does, say, in San Jose. So, and probably one of the biggest reasons is the different biomes and things like that. They've become basically acclimated to that area. So you can see the different biomes that are here. So like there's like barren land, there's like Pacific, coniferous, there's farmland, there's different things here. So if I have a case, I want to apply data, like say if I have a case in agricultural land, which is this purpley area here, I want to apply data that I've collected in that area to that area, ideally. Otherwise, I have to put something in my report saying that the data that's being collected was not from that area, but is, fairly close. And so I would put something into my report just to cover my butt. You always want to cover your butt as a forensic scientist in your report, always. So that's what I would do, and I would want to just make sure that I put that the data that I collected was from this region. Or otherwise, you know, if I collected some data from the coastal region, I would and applied it there, I would have to make sure I stated that as one of my factors in my report. And usually state several of these factors in there. And so that's a definite big factor in this forensic science, in this specific forensic science. So that was the first method, and that's one I focus on the most in my lab. Putting out pigs is typically doing successional ecology. So that's looking at the different types of insects and stuff that come to the remains. Now the body or the pig goes through a bunch of physical, biological, and chemical changes. And that draws different insects in a predictable sequence. So we use this sequence to estimate how long the body has been there for. And so I just saw something pop up in the chat. Hi, actually, the only prerequisite for this internship is FS 100W. And so, yeah, so the only so for this one here, so like if I put out remains somewhere and say, okay, let's see what happens. That's the only this is what I'm looking at to see, okay. So, and there's differences that there's lots of factors here that affect this, which is what I'm putting on my next slide, which we'll go to. Here we go. This is affected by like macro environment locations and micro locations. So like the urban environment is different from the rural environment, for example, or it could be more specific. It could be different insects come because it's in this little bushy area. It's located in this one little area. We might see different insects will go into shade. Some insects will only stay in sun. That's blowflies as well. Some insects will come indoors, blowflies included. Some will not. Some will go into a vehicle, some won't. We did some research before in trunks of cars, and of course some species will go in, some won't. We did some interesting car ones too, where we put pigs in the trunks of the car before, and did the collection of those insects. Then this was with a grad student. This was at my old school, and it was interesting because we could see, you know, what insects would come in, and it actually was interesting because certain cars had to have like a firewall between the trunk of the car and the front of the car. So the insects would come into the front of the car and move into the back, and what prevented a lot of the flies from getting there, they would get there still, but it would take some time, would be that firewall. And then we got to torch the cars, which was really cool. Okay, so and just to see what survived, and a lot of the insect evidence actually did remain afterwards, which is really, really cool afterwards too. So different factors that will affect this is, you know, are the remains buried? Are they above ground? Are they indoors or outdoors? Is the body in shade or sun? Is there clothing or not? Was the body scavenged or not? And Dr. Bolton, that was really cool. The rat scavenging the pig head was really cool. Some other factors are things like is the body, you know, hanging versus a body that's burnt? Is the body wrapped? So I want to bring up the wrapped thing because that's important, because that's a lot of things we're focusing on in my lab right now too. But before I do that, I want to talk a little bit about cases. I haven't really been paying attention to the time. Am I going over too much time, by the way, right now?
Pretty close to your allotted time, yeah.
Okay, then you know what, I'm gonna skip. Well, I should skip. I'll go really quickly. I'm gonna just skip past this here. So one of the cases I worked on was, you guys remember this show? I can't remember what this episode was called, but it was when they looked at the pig farm on Criminal Minds. Does anybody remember that one? Where they were feeding human remains to the pigs? No, okay, it looks like there's some chat stuff. Because that was one of my cases. That was my case, actually. I got to work on that case. I got to do the insect evidence on that case. So really disgusting. So I just wanted to bring that up, but I had to delete some of the slides from my other cases, but this I could sort of talk about because this case is ended now. This was it was all based on all these women who were this all took place in Vancouver. So I'm from Canada originally, and these were all basically drug addicted prostitutes who were killed by this creep. And I think my next slide shows you the creep. There he is. And he fed the remains to the pigs on his property. Okay, so he picked them up from the downtown east side when they were they were prostitutes because they were trying to feed their drug habits. And I've got these ones circled because these were the cases that I actually when I say I worked on them, my master's research actually was applied to this case and to those 6. This case was actually launched in 2001. The focus did not actually begin on him because he a lot of things were kind of ignored about this guy. They didn't think, he had anything to do with it. But it was later on that informants, there was an informant who said there was illegal firearms on his property. So they went in and then they started finding some of these women's belongings and things on the property. And that's when they finally started looking at this guy. He lived quite a 45-minute drive from Vancouver. So that's why different police force too. So they weren't communicating. And so that's how this guy got away with it for so long. He killed something like 49 women. They suspect. This is a disgusting property. So they actually had to sift through all the soil on his property to find remains. And like I said, he had been feeding them to the pigs, and the pigs basically digest most everything except for like the teeth and the hair. And so they did this by looking at these types of grids that you're seeing here on the screen. I'm just going to go really quickly now. There was a 17-month search, and by the time they got through everything, they found DNA. They found actually I think they found DNA samples from sex toys. They found 14 human hand bones from one victim, a tooth, a lower right mandible, a missing woman's notebook, some jewelry and clothing, clumps of loose hair still attached to the root, many other DNA samples, and from some of these women, they found like inhalers, they found prescription bottles, things like that. They found, and then so here's they brought in a bunch of forensic anthropology students, by the way, to help go through all this soil. So that would have been like you guys, going through and sifting through the soil to find human remains to help them out to go through this so much quicker than what they were actually able to. So I wanted to show you. So here's some of the evidence that was found in this place. And you can see. And then finally, the partial human remains of 3 victims were found in buckets, which leads to my work. So he was tried for these 6. Sorry, this picture, when I transferred over to this slideshow, it didn't go over very well. So they only went for 6 convictions because they knew they could get him on these 6. The other women, they said, we'll come back to them later at a later date. They never did, though. So they collected from the 2 sets of we collected from the 2 sets of partial remains. So what it was there was these 5-gallon laundry buckets stacked inside the deep freeze on the property. He had the skulls and the hands and partial feet of 2 of them sitting inside of the bisected skulls in the buckets. This was very disturbing to officers working the scene, by the way. Very disturbing. They were having a rough time with this. It was later discovered they both had bullet wounds to the head. And so you're looking at like some freezers on his property, because you know he had a lot of freezers because of all the meat and stuff that he had on the property. This was the freezer that where the buckets were found in. It was like this kind of thing from the court proceedings. And these were the type of buckets. These were not the specific buckets, by the way. They're just, that's what I could show you. So that's all I wanted to include about that. But like, so we worked and applied insect evidence to those cases. Now they were frozen down, the insects, but what we could show from the insects that were there was that these two women had been frozen, had been killed at different times based on the age of the insects on the body, on their bodies. One of them had been must have been out and exposed to insects for a lot longer than the other one, for months, compared to the other one who would have been exposed for maybe weeks. So we do that kind of casework, plus we do child, senior, and animal abuse cases. For me, this is the absolute most disturbing casework that I've done. Because they're alive still and they're living with insects on them. So this here has been my absolute worst casework, that I just can't stand doing this stuff because they're alive still with insects on them, and they can't, you know, defend themselves. They can't, you know, look after themselves. So this is what bothers me the most when I do these kinds of cases. I'm not going to go into more detail than that about that, but just so you know, that's what bothers me. So I wanted to leave cases like that. And just quickly go into more about the internships. What you guys I'm going to skip this one because it doesn't need to be done. So for internships right now, I've got everybody working on looking at delayed colonization of concealed remains. This is a big one. Working in cases, we discover remains all the time that have been concealed in something, whether it's in a box, it's wrapped up in a rug, in plastic, whatever it might be. A lot of people do this to remains, whether it's to avoid discovery, for cleanup purposes, for disposal and cleanup, to move the remains because it's easier to maybe drag remains in a rug or something like that, or just to hide the remains for remorse or regret. So they'll, dump the remains somewhere and it's wrapped up in something, or it's in a box, or it's in a suitcase. And I've had this very type of case before. So I just wanted to just look at this now. This kind of thing can delay insects from getting to remains. So the point is, now we're looking at how long is that delay? So that's what my students are working on, and what I would continue doing with you guys into the fall, because we need to do more of this stuff still. So in the field, we do research on pigs mostly. In the lab, though, it's beef liver. And we've shown with publications and so forth that beef liver is excellent for doing representative work for human remains and stuff, because it is very representative. I've even published stuff on this. So this is part of the lab. So what you guys would basically be doing is putting something, like you would conceal the remains in something, and we'd do 5 or actually 4 experimental we've got 4 experimental cages, and a 5th one would act as a control. And we put them into this chamber, and we've got the chamber set for a specific temperature and lighting, and then we'd alternate the lighting so that it follows more of a day pattern. And basically what you're doing is you are checking those all the time to see, has there been any egg laying? Have the insects laid? And what kind of, have they moved into the boxes yet, or into the, have they reached into the shrink wrap, or have they gotten in anywhere yet? So we're looking to see what is the delay pattern here. And that's what the internship has really been about. We're looking at dog waste bags as miniature garbage bags. Hated doing this one. really, really stunk for some reason. And it was bad. So we will probably come back to that at some point, but I didn't like that one, but we'll come back to it. But like here's like the miniature cardboard boxes. So this was done with the first species that I did. I think it was Calliphora latifrons. And then we're doing Lucilia sericata. So this is what I want you, this is what's happening this semester right now, and hopefully continue on to maybe another species in the fall. And so the cardboard boxes are taped shut all the same way. And we're looking to see, they like to go into the bottom, if the flies lay the eggs at the bottom of the boxes, and they try to sneak in usually where there's some blood that's dripped there. So we have to look also at boxes where the body's not bloody, and so maybe might be a little bit of purge. And so that's what you're looking at. So we label the boxes and so forth and try looking at experiments that way. And suitcases. Now I can't get little miniature suitcases. I've tried my damnedest to get them. So we're using little makeup bags that are like fabric, sort of like a fabric suitcase with the double zippers. And you can see they've laid eggs along the seams. That first species, they did not get in at all. So we find that if there's maybe a gap, somewhat of a gap in the zipper, that's how they'll get in. That's the only way that any insects get in. And we have to make it so it's small enough that the adult fly doesn't get in, but that the hatched eggs, maggots will get in. And then shower curtains. These are cut shower curtains with beef liver inside of them. We've tried different methods of like if they've been duct taped, and how well they've been duct taped, and that sort of thing as well. Shrink wrap, they have no problem getting into shrink wrap. Just remember that too, by the way, when you have a shrink wrap sandwich or something sitting out. Flies can get in there, lay eggs, no problem. Disgusting. Still to come, I need to do rugs. I need to do wrapped up bodies and blankets. I want to do hard shell suitcases, an example like this sort of thing here. And do an outdoor comparison, like lay out like suitcases outdoors, blankets, rugs, whatever, with something inside of it, like pigs preferably, but probably not on campus grounds.
Is there any smells or adhesives that the bugs don't like that could prevent them from going near it, or does the smell of what's rotting overwhelm all that?
It depends. For the most part, people have tried spraying things, but pretty much they will go. There's no real, like people will try to use like bug sprays and stuff to try to keep the insects away and stuff. It doesn't really work that well. It doesn't work that well at all, actually. So they might be able to like kill the flies that are there at the moment, but they will come at some point. So yes, there's been research on that very thing where they'll spray down body or they'll douse a body in something like kerosene or whatever it might be to try to keep the bugs away. So yes, there is certain things that can work that might not work or might work for just a short period of time. This was some of the findings that I had found before that, the eggs are laid in like the seams of the cardboard boxes. For the dog waste bags or garbage bags, they may be treated. I'm not sure if they are treated or not because so I wasn't sure because the flies they'd lay in the open control bag for sure, no problem, but they didn't lay they wouldn't lay on the bag, like with the bags itself, like when the bags were knotted closed. That was for Calliphora levita. And for the fabric suitcases, they had eggs laid on the zipper. They hatched, but they did not survive beyond the second instar. And so these are just certain things that some of the findings that I was getting. And this is what I want to do now. So some of the things we really do focus on, I think this is one of my last slides here, is that lab safety and would focus on life cycle, colony, and rearing, prepping for experiments. You guys are going to run experiments. You maintain experimental insects and colonies in the lab. You would submit a final research paper, plus a journal of what was completed over the semester. And that's pretty much what the internship involves. And I think that's pretty much all I need to cover with you guys. I think this is nothing. Yeah. Oh yeah. That's just, yeah, I can skip it from there. I'll leave it from there.
Thank you, Dr. Warren.
Thanks.
All right, next we have Ian presenting about a digital forensics internship.
Yes. Hello. Thank you. Can you guys hear me okay?
Yes, we
Cool.
can.
Thank you, Jodie, for that awesome
Oh.
presentation. Definitely more interesting than mine. Let's see if I could pull it up here. And you guys are able to see that, yes? Cool.
Yes.
All right, so just a little bit about me. What's up, guys? My name's Ian Obniana. I am a recent graduate here at San Jose State with the Digital Evidence Program last spring, where I completed my internship under Bryce Westlake. He may have presented earlier in this meeting, but that was my internship coordinator. And throughout my internship, I gained valuable experience with digital evidence. And following my internship, Bryce actually had offered me the opportunity to step into the role of the lab manager here at the laboratory we've established at SJSU. And as lab manager, I kind of oversee the day-to-day activities within the lab. And I mentor a group of interns, teaching them curriculum on digital forensics, as well as the software and workflow we have in our laboratory. So what is digital forensics? Sorry, so about a little bit about our lab. Sorry, I skipped a slide there. So here's some background of our lab. So SVDFL, so Silicon Valley Digital Forensics Laboratory, is a digital forensics lab that does research, and we are an investigative lab established through SJSU, law enforcement, and government agencies. So San Jose State University Police. So we do have a MOU partnership with the San Jose State University Police here at San Jose State. And we work with them on day-to-day with devices that they bring to us if they have a case. And we provide students with real-world experience. So students do get to work on these live devices that UPD does bring us, and they work directly with that evidence. So interns, yes, they do get hands-on training and evidence processing, forensics analysis, and investigative techniques. And our program in the laboratory is officially approved internship site for JS 181. So you can use this internship for that requirement. And it gives you also that practical experience. Okay, so what is digital forensics? Awesome. So digital forensics is the scientific process of recovering, analyzing, and reporting on digital evidence. Digital evidence being mobile phones, it could be an iPad, the new Ray-Ban glasses that came out, VR, a TV, security cameras, anything, the ECU for a car, it could be anything like that. But our laboratory mainly focuses on mobile device collection. At the laboratory, we use a variety of industry standard tools. One of our main tool being Magnet Axiom. So this tool kind of sifts through and filters all the data within that device, making it easier for the practitioner to look at all the data and find what we need, evidence that we need, filtering all the unnecessary out. Gray Key is another thing that we use and recently acquired. Gray Key can access phones. So if a phone is stolen or lost and we have a phone that we don't know the password to, we can use Gray Key to crack that password and gain access to the phone, whether that be a lost phone or a suspect's phone. And Autopsy is another open source forensics software that we do use. Interns will learn how to use this software, and they collect, analyze, and interpret this evidence. Okay, interning at the laboratory. So what is it like to intern at the lab? Interning at the lab is a very unique opportunity to gain real forensic experience while you're still in school. So I was able to get that opportunity, and it was great. Unlike classroom learning, you'll be working on real cases, not example cases. So these are actual digital evidence, and you'll also get hands-on experience using recent cutting-edge forensic tools for digital evidence, which is a great advantage when you're applying to jobs, as you already have some experience working with these tools that industries today that are already using. And beyond casework, interns have opportunities to contribute to research. So we do also have a research side of our internship. So you can either do a hands-on portion, or you can do a research. If you don't want to go hands-on with actual evidence, you can sign up for research within devices, digital evidence. So research on how can we change settings on the phone to make it better for us to find more data. How should we have this phone initially when we get it? Should we leave it in the state that it's in? Should we turn it off? Those kind of things, those kind of research for progressing digital evidence. One interesting one that I recently learned about was that you can actually freeze the RAM on your mobile phone. So actually putting it into a freezer and making it cold for about 15-20 minutes, and that actually slows down that data transfer within that RAM and alternating data, which gives more evidence for us to look at, essentially. Okay. So some intern responsibilities. So at the lab, interns play an active role in forensic investigations. Like I said, you guys are really going to be dealing with real evidence. And your key responsibilities include learning and applying forensic imaging and acquisition techniques, handling and documenting digital evidence, conducting preliminary forensic analysis, and then maintaining chain of custody and ensuring proper evidence handling. So interns will be going through the full cycle of digital evidence. So from the intake all the way back to the return. And they will follow procedures accordingly. And these are critical skills for forensic professionals and will prepare you for roles in law enforcement, cybersecurity, or even private forensic labs. Okay, some skills gained. So by the end of your internship, you will have developed core forensic skills, including proficiency in digital forensic tools like Axiom, like I said earlier, and the other tools. Another skill is increased critical thinking and investigative skills. So as you are kind of sifting through this evidence and filtering the data, your critical thinking and skills will naturally gain. It'll be easier for you to find what is needed. It won't take as long as before. You'll know exactly where to go. And you will gain proper evidence handling and chain of custody procedures. So understanding the integrity of the chain of custody and how important it is to maintain that integrity. And also learning how to create forensic reports and writing and documentation. So at the end of your acquisition here, you will be writing reports. There will be a summary report, which kind of goes out to the agency that submitted the phone. So a summary report would be a quick kind of report of what you did, what you found, and that would go back to, for say, University Police, so UPD. And then there's a technical report where it's a full step-by-step write down of what you exactly did with that device. And any hiccups that you may have ran into, anything like that, all of it should be documented. And then these skills make you stand out when applying to different jobs in the digital forensics field. When you're applying and they ask you, what did you actually do with these devices? You can actually explain what you did. And you could say how many devices you've done, which is great. Okay, yeah. So some internship requirements. So to qualify for the laboratory, you must meet some You must have completed FS12, so that is our intro course to digital forensics. You must commit to 2 semesters, so it's a 1-year minimum internship. And you must pass a live scan, fingerprint, and background check. So live scan is easy, you just get your fingerprint, but a background check is needed. It is the same level background check as the cadets at school. So the police cadets that you guys see around school that are driving the carts around, I think they wear blue. So they go through a background check that is the same level. This is so you guys gain access to where our facility is. So I'm currently in the laboratory, which is located in the south garage, where the parking service is on the 3rd floor. So this is a secure building, and UPD operates in this building, so background checks are needed. Lastly, must have a strong attention to detail and confidentiality. I guess to note, if you don't want to go through this full background process, you can opt for the research portion of the internship. So you don't need to be background, as you'll just be doing strictly research. But if you want to work hands-on with the real digital evidence, you do need to have a background check. Yeah, this is because it's just a serious responsibility, and we need interns who are ready to learn and committed to being a professional. So the application process, here's how you apply. So applications open twice a year, so November and July. You can submit your application on our website, or you can submit your applications directly to me, and you'll get an interview with us. There's a kind of like a 2-step process interview. It's not anything crazy. We just want to get to know you. We are currently accepting digital evidence and CSI students only. They're given more priority as the chemistry students are a little bit irrelevant to what we're doing here. But we are welcoming the CSI students, but prioritizing the digital evidence. It is a very competitive process, so it is good to highlight your interest in digital forensics during that interview. So internship progression and promotion. So in the lab, interns progress through structured training phases designed to build their forensic expertise kind of step by step. So phase 1, would be semester 1, you're a trainee. This would be your learning phase. So where you will be introduced to forensic processes, lab procedures, and digital evidence handling. And at this stage, you won't work on live cases, but you will focus on training and skill building under my supervision. So phase 2, semester 2, you'll become a junior technician. So after passing your background check and completing initial training, you'll start working on real forensic cases, so real live evidence, again, under my supervision. And you'll apply what you've learned in that first semester with the evidence acquisition, forensic imaging, and kind of case analysis. So post-internship, phase 3 here, you become a senior technician. And if you continue beyond your internship, you can be promoted to this position, where you'll take on higher level casework and mentor the newer interns coming in, so junior technicians or trainees, and assist in lab operations like tool validations here, like performance verifications and basic maintenance around the lab. So there's some advancement opportunities at the end of our internship. So senior technicians may be eligible to become certified with the main tool that we use here in our laboratory, Magnet Axiom. So you can have eligibility to be certified, and even potential part-time employment at the lab. This progression just ensures that you guys kind of build that real world experience and skills. So when you're applying to jobs, you already have that experience, and it's easy for you to say, I've done this, I've done that, I understand this, I know how to do this, I'm efficient in this. Okay. Okay, research opportunities. So beyond casework, this is the kind of the research portion of the lab. Interns can participate in federally funded forensic research projects. They can also participate in student RSCA fellowship and CSU research competitions. as well as there's other opportunities to collaborate with our faculty like Bryce Westlake and for forensic research. Also, if you're kind of interested on the academic side of forensics, like teaching, this is a great opportunity for that. Okay, so some career pathways after the laboratory. So interns who complete this program can go into working into different law enforcement or forensic labs, as well as some cybersecurity firms. Some potential career pathways also include three-letter agencies like CIA, the FBI, the DEA, the DOJ, the Department of Justice, and as well as San Jose State PD and UPD. Private forensic labs are also hiring for this after. So one like Celebrite, even Magnet Forensics, and even cybersecurity companies like Cisco and IBM offer digital forensic positions. There are private labs that do legal and corporate investigations. So it's more like e-discovery. So if you don't, if you don't want to go into more of the criminal side of digital evidence, there is different kind of areas. There's e-discovery, which deals in like legal litigation, where say you're not trying to find a crime on a phone, but instead you're trying to find some information that a person needs in their phone. They're digging through it, looking by themselves, but our tool can find that really fast. And again, these are some just great skills you can gain at the lab. All right, I think that's it for me. So here's a contact for us. So my email is there, as well as Christine Martinez. She's our technical program manager. Most contact information will go through her, but me and Bryce do reply. Bryce is our laboratory director, and his email is there. Our website is posted there, and as well as our location. Any questions? If not, then I think that is it for me, guys. Thank you so much.
Thank you, Ian.
Awesome.
All right. Next, we'll be hearing from Everett about the record clearance project.
Well, good afternoon, everybody. Thank you so very much. Let me just get this all set up here. And okay, let's see. Folks, are you able to see my screen okay? Are we able to see the PowerPoint in its full mode here?
We are. Thank you.
Thank you very much. Fantastic. Well, thank you so much for having someone from the Record Clearance Project come and share a little bit about our different opportunities, but I think this will be of interest to some folks. My name is Everett Glynn. I am the co-teacher of the classes that the Record Clearance Project offers. And I'm going to be sharing a bit today about what our program does. And I know that this is a forensic science club gathering. So I tried to make this presentation a little bit about, what opportunities we have, but also just a bit about something that, folks from all over the campus find to be an interesting thing that our campus offers. So what exactly is the Record Clearance Project? We are a clinical law firm. So we are a actual nonprofit office. We do a variety of different legal services for folks across the state of California. Oh, thank you very much for that, Nikki. Yes, and I have a couple things I'll be sharing as well. That's great. So we partner with San Jose State University and offer several classes. They are upper division college courses here, which I'll go into in a little bit. But before we are college classes alone, we are actually a full-on clinical law firm. We have supervising attorneys, and I like to set it up kind of like this. If you were to look at folks that are in law school or even like med school, what you would see is you have the students and you have the the students going into the room with people that are, the patients, and the students perform a analysis. They try to figure out what's going on. They come up with something like a diagnosis, and they would go back to the supervising doctor and they would propose their diagnosis. And then upon receiving the doctor's approval, they would go back in and treat the patient. In law school, there's something similar. Law students will have the opportunity to practice law and actually provide legal services prior to actually being, California bar certified attorneys. This is the only program in the country, as far as we know, that does something similar, but for undergraduate students. And so that's what we'll be talking about today, the Record Clearance Project, which provides the only undergraduate clinical legal program that we know of in the country like it. So what does the Record Clearance Project do exactly? A lot of people would think about um, the system of incarceration. That's something that even if you're, not really in the justice studies where we really dive into, criminology, the study of prisons, folks would be aware that we have 2 million people in prison, several million people on parole and probation. What a lot of folks don't quite realize is that 1 in 3 people in the United States have some form of a criminal record, whether it's an arrest, a misdemeanor record, or felony record. And so, of those three things, 1 in 3 people in the United States have some form of criminal record. Over 20 million people have felony criminal records, tens of millions have misdemeanor records. And again, when we factor in arrests, it's 1 in 3. That would be one person to the right, one person to the left. Now, what's the problem with that? Well, they have a lot of studies that show that people who have felony level criminal records experience up to a $480,000 lifetime income depreciation from someone who does not have that felony record. And that even goes for people who didn't even go to prison for that felony record. Even someone who just received a felony, took a plea, didn't receive a custody sentence, they could still experience lifetime income losses of upwards of $100,000. Now, why would that be? Well, the last presenter, Ian, just as a kind of a little note, he had mentioned, the background process. Well, 1 in 3 people have a record, and 9 in 10 of employers require a background check. And the fun fact about record clearance in California is it broadly allows the clearance of those criminal records, those arrests, those misdemeanor, those felony records. And what that record clearance does is, by law, for those private employers, it would withdraw the ability for the state to disclose those records on background checks. So if I was to have a background and I have these dismissed records, by law, they're not supposed to appear on these, and they're not supposed to affect the hiring process for me. So all of a sudden, those 9 in 10 jobs that are screening people, and they're perhaps dismissing applicants based on the presence of a record, when my record is cleared, that would no longer be an issue. For government jobs, it's a little different, but most government jobs, including, contracting, jobs like if I want to be a nurse, they won't let people in even with like a misdemeanor level record. The Record Clearance Project, we clear those records, and then now people would be able to access those jobs. As a little fact, there was a study done by Stanford's Public Policy Institute. They looked just at our program specifically, and they found that after our clients had their records cleared with our program, their income went up the next year on average by about $6,190 in 2014 dollars, which is like over $8,000 today. And so here's the really cool thing about record clearance in California. It's really broad. So the problem with that is that not a lot of people are able to access this, frankly, life-changing service. We try to change that, and we use students. Students do all of our work, and this is where it gets kind of cool. So the students, and so I'm a teacher of this program now, but I actually started in our beginning classes that I'll get into in a little bit. But what the students do is, so they're under attorney supervision, and they are trained to provide the legal services practicing under the bar card of the attorney that supervises them. So just like that medical school thing we talked about, just like the attorneys that are the law students, the students in this program are going to provide 3 really important services that we'll talk about kind of how they work and what students in these classes are going to be doing. The things that we do we take the record clearance law, and just kind of a fun fact, just think like, what do I know about record clearance prior to this presentation, or maybe prior to coming to this university? The fun fact is we find that about 90% of our clients had absolutely no idea that these services were available prior to talking to us at the services that I'll be getting into. We educate, we provide individualized legal services to people who need to have their records cleared, and when it's necessary, we will represent people at court to make this happen. So what we have done is when people start taking the record clearance classes, the first thing is there is a beginner course, and they're going to be practicing and training these 3 services as well as applying them. Then there's the internship where they're going to be doing the court representation and actually able to advocate for clients in court. The first service that RCP provides is we have presented to over 16,000 people across the state, including over 5,500 people who are currently in custody. And we go out into the community and we educate people about their legal rights related to getting their records cleared. Kind of a fun fact is as of January 1st of 2023, almost all California state prison cases became eligible to be expunged from someone's record. The unfortunate reality is very few people were informed of that. So we often have people being told by their parole officers, their probation officers, police officers, their friends, their family, your prison case wouldn't be eligible to be cleared, so that's always going to show up on those background checks, bar you from employment. We get the opportunity to go out and explain to people that their cases are eligible for record clearance. Oftentimes, from these presentations and across a broad range of organizations that the Record Clearance Project partners with, we host and provide brief legal service interviews for anyone in the state of California if they want to sit down with our team. They want to go over what appears on their criminal records and the next steps in the record clearance process. A fun fact is about, I would say, 60% of the people that we talk to in custody believe they are convicted of something more serious than they actually are. And so what that means is when I'm arrested, I oftentimes think I'm arrested for a felony because a police officer has the authority to charge me with just about anything they want. I end up getting convicted at court of a misdemeanor, but I'm living with the perceived stigma that I have a felony on my record. At these legal service interviews, the Record Clearance Project will review what appears on someone's record. They do this by looking at something called a rap sheet, the record of arrests and prosecutions. We'll look at those reports. We'll look at what appears on their record, and we will provide advice on the next steps in the record clearance process. Now, earlier I mentioned that folks tend to see about a $6,000 average increase in their income following record clearance. Well, we're pretty versed on the laws as they relate to record clearance, but specifically the informal practices in Santa Clara County. What we find is that about 50 to 66% of people who meet with us at these brief legal service interviews have their complete records cleared after one conversation with us. And so we're thinking about all of the disadvantage that comes with those records cleared after one meeting. Some people will require court representation to have their cases dismissed. In these cases, what happens is our student teams will work with the clients. They will prepare the documents necessary to go before a judge and request that those cases be expunged from their record. As far as we know, RCP students are the only undergrads in the country that are able to actually speak in court on behalf of their clients. Why is this important? Well, a lot of the times we're seeing a lot of old cases just dismissed from people's records. So a lot of our work is just letting folks know what those next steps are going to be. But as of January 2023, as I mentioned, a lot of cases are eligible to be dismissed. So we're seeing a lot more of this need for people to be representing clients in court to ask the judges to dismiss these more serious cases. And these 3 steps, this is where the students come in. So I'm talking about this stuff. I'm getting into things like this is the financial benefit. The American Bar Association, just as a little fact, they did a kind of a survey of daily life in the United States, and they found over 44,000 barriers that someone with a record will experience if they're just trying to navigate life. Some of the interesting ones that, like, you wouldn't even imagine this. You want to join a parent-teacher association, not with a record. You want to get student loans, not with a record. Immigration, if there's felonies on that record, those felonies would have to be reduced to a misdemeanor to protect people during the immigration process. There is so much that record clearance can provide, and the students through this record clearance project get the opportunity to provide these services, practicing the law under the license of the attorney. Now, how do we learn about all this? This is where students would get to enroll in an introductory course that would train students on applying record clearance law. In addition, they will provide the legal services. So we're not so big on tests in this class. We take tests to practice, but we don't really grade based on tests. What we look for is we want to actually practice this with real clients. And so we train. We train as if we are receiving employment training, as if we are, under the same ethical standards as an attorney. And when we sit down and we provide these legal services under attorney supervision, we are acting under our attorney's bar license. And so in our JS 140 class, folks that enroll in this class, it's a 3-unit upper division course. And so if folks are interested in this, this could be an elective course. And so this would be the first semester. Students are going to learn how to do legal analyses. So they're going to learn how to analyze these rap sheets. They're going to get a ton of very practical training on the criminal justice system. Everything from how sentencing works to how to provide legal information, how to do legal research, legal interviewing and writing. You're going to get training from clinical social workers on how to do trauma-informed interviewing. So we work with people every day who have experienced trauma. And so we train pretty extensively on this, but even in this first semester, folks will get the opportunities to provide these legal services, review people's records, assist in the record clearance, all during this first semester. So this is our introductory course. We have a lot of folks come from all different majors to take this. We have computer scientists. We'll have people from different majors in the social sciences program, engineers. People really like this program because they get to use their education, even in this first semester, this beginning 140 class, they get to use their educations to give back to their community by providing these legal services. This legal service interview I mentioned here, I met with someone the other day and provided one of these for them. He told me that he had done a similar review with another office that charged him $3,500 for the service. In order to have his record cleared, he was going to need to work with them for an additional $6,000 to get the record clearance relief that he needed. It took me 30 seconds to fill out a form for him that ended up dismissing everything that appeared on his record. And so students in the 140 class really enjoy getting to give back in that way. And for me, I was on track to be in a totally different field, but when I was in the 140 class, I realized that this is the kind of legal work and the kind of public service that I want to be doing. A lot of folks, after taking this beginning 140 class, some take it for an elective, some take it then graduate. We also offer 2 different internship opportunities that are also 3-unit upper division classes with the university. So by the time someone has finished an internship with the record clearance project, they would have 6 units of upper division coursework, as well as their internship complete. The first internship we offer is our legal services internship. So what this is this is going to be your training in that first semester. You're providing legal services with often advanced students or program volunteers. You train in the first semester. You apply the work in the second semester. You will actually represent and you will work on representing clients for clearing more serious cases in court. And so you would do this under attorney's supervision. You'd have a partner. Our legal service interns right now, they're all working on cases to appear on calendars this semester. So they'll be going before judges and they will be with their clients speaking on their behalf. They've prepared all the documents. They've prepared the petitions that explain to the judge why these cases should be dismissed from the records. March 24th is going to kick off a week where our legal service interns are going to be training folks from Stanford Law. So Stanford Law will send us students who will work with our program for a week. So SJSU's undergrads are going to spend the week of March 24th training Stanford law students how to use record clearance law, and then they're going to be working with clients and doing an accelerated process to train Stanford law students to practice record clearance law. So kind of a cool thing on the resume after working with the legal services program is I trained Stanford law students when I was an undergrad. So that's going to be our legal services side of the program. The record clearance project as a whole partners with another organization. We're the same entity. We're both parts of the record clearance project. And it's going to be our mentor program, our services navigation program. This program is going to be a mix of people who were formerly incarcerated themselves, that the RCP hired to serve as peer mentors for people that are just being released from custody or are in particularly precarious economic or social situations. So people that are unhoused. Oftentimes we'll be working with people to clear their records, and they may be currently experiencing housing insecurity or food insecurity. And so what this service navigation program, this mentor program does is we pair folks with one of these mentors who have successfully done reentry from being in custody, and they will work with people who are, you know, just being released to kind of, it's, I don't know if anyone here has seen kind of how reentry works, but it's very much a patchwork of social services and a lot of paperwork, and folks won't have things like an ID or a social security number. They won't have somewhere to go. They won't have food to eat. And so the peer mentors there, it's their job to make sure that the needs of the people that are reentering are met so that they can successfully get away from the criminal justice system and toward stable employment, stable housing. We call it our path to expungement program because what happens is people are released from custody and we work with them all the way to getting their record cleared. And so some of our interns end up working alongside these peer mentors, and so they would help with the they might provide some of the same legal services for these folks. They will go into custody. They'll meet people in custody and start developing those plans. So when people are released, they have places to go. They have their needs met. They have their basic needs met. Um, and this program broadly assists people with their reentry needs. And so our interns in the JS 142 class have the opportunity to work alongside the fabulous peer mentors in this program. So just as a quick recap, we have the beginning 140 class, that's learning about the record clearance law, getting practical training on the entire criminal justice system, learning about how sentencing works, how to do legal research, how to work with people who are experienced trauma, how to be a trauma-informed interviewer. In a practical setting, we work with real clients in a controlled environment, so folks are always paired with people who have done this before. We segue into these two internships, the legal services side and the service navigation, the peer mentor side. And so how do we start this process? Well, the JS 140 course is going to be a prerequisite to taking either JS 141 or 142. So it ends up being a 2-semester process. Oftentimes what happens is our students in the JS program will take JS 140 as an elective alongside a 1-unit co-requisite. It's like a lab. So our JS students are probably less familiar with like the 1-unit lab classes as folks in the forensic science program. But there's a 1-unit co-requisite to practice some of that legal research and some of the other practical trainings that folks receive in the classes. And so what happens is you would take the JS 140 class in the first semester, and then the admittance to the JS 141 or 142 would be just for folks that did well in the 140 class. This is where they want to be for an internship. And they have satisfactorily completed the training in 140. We are held, when we're students in this role, we are bound to the same ethical obligations as attorneys. So there's competency that has to be met there, but for the most part, folks that are driven in this program end up having no problem joining the internship after the 140 class. So as a roadmap, if I was to take JS 140 in the spring, I would be looking forward to taking the JS 141 or 142 in the fall. And so I'm going to talk a little bit about the JS process here and other majors as well, but I want to kind of open up with a sort of disclaimer here. I was talking to Dr. Westlake, who's one of the teachers in the forensic science department, and I don't know, I think maybe all of our forensic science professors left for the night, but I would highly recommend if folks were interested in taking either JS 141 or 142 as a possibility of satisfying that internship requirement for the major specifically, please check with an advisor to see if that's okay before doing it. I was looking at your major forms prior to this presentation, and I didn't see JS 141 or 142 listed as satisfying the internship. So please, folks, if folks were interested in the two class, the JS 140 and the JS 141 or 142 internship options, please check with an advisor in your program, see how those two courses are going to fit with your roadmap prior to registering. That said, folks are welcome absolutely to take the JS 140 class as an elective if it fits their schedule. We welcome folks from all majors. We've had folks as far as music and political science, communications, engineering. All are welcome in our 140 class. It's a really great experience. You'll get a lot of practical insight into the justice system. But if folks are thinking of satisfying an internship requirement from there, please sit down with your advisor and make sure that's okay before doing so. I've heard that folks have been able to use this as their internship in the FS majors in the past, but I can't stress this enough. Please check with an advisor before building this out into your 4-year roadmaps. All right, and that's all I've got for today. So thank you again for having us. We really appreciate it. Folks are always welcome. We do have events all the time. If folks wanted to be in the loop about some of the stuff that we're doing, I put my email here. If anyone was interested and had any questions, including things like, hey, how would this fit into, this major? I think there are maybe a couple non-FS students that will be attending this. So if folks were interested and had any questions, I'll put it in the chat right after I stop sharing my screen. I'll put our email, and we also offer services, as I mentioned, to anyone in the state of California. So, as I mentioned, 1 and 3, hand out to the right and left, one of the shoulders that your hand is on, they may have a record. So if folks were in need of record clearing services or know someone that does, they can also get in touch with our program as well. So that's all for me. Thank you very much, folks.
Thank you, Everett. All right, finally, we have Mary Juno who will be presenting about Themis and the Scotland trip. She couldn't be here in person, so she sent the recording, and I'll go ahead and share my screen for that. Here we go. Okay. All right, can everyone see my screen okay?
Yep.
Perfect. All right, let me go ahead and play the recording.
Hi, folks. How are you? Juno here. I'm sorry I couldn't be with you live for this event. It happens to be my best research trial. Here's one. So, what we're looking for, yes, So.
Oh, sorry, can't hear the audio. Didn't realize that.
I do.
Not gonna find.
Alright, let me see if I can try and do something about the audio. Can anyone hear this?
I personally cannot hear anything. When it was playing, it was coming in and out, but not consistent audio.
Really weird. I'll see if I can do anything on my end. But if not, I can go ahead and send the recording to everyone. Because I'm not sure what I can do on my end about it. Let me just go ahead and send out the recording. And I'll also put in Juno's email, so if anyone has any questions, they can reach out to her about that. Sorry about that. I think that will conclude it. Thank you guys for coming. Sorry about the whole thing for Juno's presentation, but we hope you took something from this presentation, that you learned about the opportunities, and we hope to see you at future events. So thank you for coming.
Thank you so much. Thank you for organizing this.